Hello Prof. Annie, my name is NURHAZWANI binti Jefri AZWAN, and today I'll be presenting my poster, and the title of my poster is Acyclovir, an antiviral drug that has played a key role in the treatment of herpes infections. So let's first begin with its origin. So Acyclovir is a guanosine analog, and it is primarily used to treat herpes simplex, varicella zoster, and herpes zoster infections. This drug was first developed in the 1970s by the Burroughs Wellcome Company. Behind its development was the remarkable scientist Gertrude Elion, who later won the Nobel Prize in Physiology in 1988 for her contributions to drug development. Eventually, Acyclovir was approved by the FDA on 29th of March 1982, marking a huge milestone in antiviral therapy. Now, let's look at its structure and properties. So, Acyclovir belongs to the class of guanosine nucleoside analogues, but it has an acyclic structure which makes it unique. Its IUPAC name is a bit of a mouthful, which is 2-amino 9,2-hydroxyethoxymethyl 6,9-dihydro 3H-purine 6,1. Its molecular formula is C8H11N5O3 with a molecular weight of 2 to 5.20 gram per mole. And Acyclovir appears as a white crystalline powder, and it is soluble with a solubility of 1.4 mg per ml at 25 degrees Celsius. However, its oral bioavailability is quite low, around 10 to 20%, which can affect how much of the drug actually reaches the bloodstream. Now let's move on to the mode of action of Acyclovir. So how does Acyclovir work? The story begins with the herpes simplex virus, or HSV1, where the drug shows one of its best effects as shown in the diagram above. Acyclovir is a guanosine analog that mimics a DNA building block. But what makes it clever is its selective activation, where it is only activated in virus-infected cells by an enzyme called thymidine kinase, or TK. Then, TK converts Acyclovir into acyclovir monophosphate, ACVMP, and then the host enzymes convert it further into acyclovir triphosphate, ACVTP. This active form competes with DGTP, which is a natural DNA building block during viral DNA synthesis. Once incorporated into the viral DNA, Acyclovir lacks a 3' OH group, which causes premature chain termination. In simple terms, the virus cannot finish making its DNA and replication comes to a halt. Additionally, Acyclovir inactivates viral DNA polymerase, further preventing the replication. After knowing the mode of action for Acyclovir, now we will be moving on to resistance. So, as with many drugs, resistance can occur. The most common cause of resistance occurring about 50 to 80% of the cases comes from mutations in the UL23 gene, which codes for thymidine kinase. There are different mutation types. Some viruses don't produce thymidine kinase at all, and others produce thymidine kinase with reduced or altered activity, affecting Acyclovir activation. These mutations often occur in GC-rich regions of the genome. There is also another gene called UL30, which encodes DNA polymerase. Mutations in this gene are less common but still important, whereby they can interfere with Acyclovir being incorporated into DNA or even remove Acyclovir after incorporation. So moving on to the course of treatment, in Malaysia, Acyclovir Stada 400 mg tablet is priced at around RM25.73, while the Acyclovir Stada 5% cream 5 gram costs about RM18.75. These are relatively affordable options considering the important role they play in managing viral infections. To wrap up everything, Acyclovir has revolutionized the treatment of herpes-related infections. From its discovery in the 1970s under Gertrude Elion's guidance to its modern-day use, it stands as a powerful antiviral that works by targeting viral DNA synthesis with remarkable precision. Even with emerging resistance, Acyclovir continues to be a cornerstone therapy in antiviral medicine. Thank you for listening, and I hope this presentation has given you a better understanding of this amazing drug.